Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-level biology topics. This is specific and non-specific immune response to infection. Topic CB1, non-specific immune response to infection. Beginning with cell recognition. Cells have surface molecules, and examples of these are glycoproteins. These help them to bind onto each other, and this allows them to be held together in tissues. Also, they can use these surface molecules to distinguish between self from non-self, meaning immune cells can be able to distinguish between body cells as well as foreign cells. Non-self glycoproteins, or the antigens, are going to be different from the body cell proteins. Antigens are going to be recognizable compounds or chemicals. They include proteins, glycoproteins, and sometimes they can be carbohydrates. Antigens can also be toxins from specific bacteria. Antigens can be toxins from specific bacteria as well as viruses. Like I said already, antigens can be recognized by the white blood cells. This sometimes is called leukocyte with a K or leukocyte with a C, and this occurs during specific immune response. Non-specific immune response is going to be initiated by any pathogen, while specific immune response is going to be specific to a particular pathogen. Looking at the specific leukocytes or the immune cells, these are going to be involved in the body's response. Some of these are going to be specific, while others are non-specific. Leukocytes are produced in the bone marrow, and they mature in the thymus gland. They all contain a nucleus, and they have callous cytoplasm. These leukocytes include granulocytes as well as agranulocytes. If we can see here, the agranulocytes have no granules. As you can see, they do not have those tiny particles, which are the granules. These include the lymphocytes. Lymphocytes are the ones that produce antibodies. They include the monocytes. Monocytes are the ones that become macrophages that participate in phagocytosis. We have the basophils, neutrophils, as well as eosinophils. These participate in non-specific immune response, while these ones are more involved in the specific immune response. Beginning with the granulocytes, these have granules like we've already seen in their cytoplasm, and these granules can absorb stain, which can be viewed under a microscope. They have lobed nuclei with projections, and again you've seen here, they are lobed like that. They have projections, and they are mostly involved in the non-specific immune response to infection. These include the neutrophils. The neutrophils participate in phagocytosis, and they engulf and digest the pathogens. They are multi-lobed nuclei with 70% of the leukocytes being neutrophils. They have many lysosomes, and these lobed nuclei allow them to squeeze out of blood capillaries into specific areas where infection could have spread. Other granulocytes include eosinophils. These are also involved in non-specific immune response, and they participate more in response against parasites, allergic reactions, as well as during inflammation. These can be stained red by eosin stain. The basophils, again still in non-specific response, these are two-lobed nuclei. They produce histamines, and these histamines again are involved in inflammation as well as allergic reactions. Moving on to agranulocytes, these do not have granules in their cytoplasm, and they do not have lobed nuclei. They are mainly involved in the specific immune response. They include the monocytes, like I already said. These are the largest of the leukocytes, and they pass through tissues to become macrophages, and these participate in phagocytosis. The lymphocytes are also leukocytes, and these have very large nuclei. They participate in specific immune response because they become B cells as well as T cells. Non-specific immune response to infection involves inflammation, in which the mast cells as well as basophils produce histamines. It includes fevers, and these fevers prevent pathogens from reproducing effectively, so the higher temperature lowers the reproducing rate or the reproduction rate of the pathogens. There is phagocytosis, in which digestive enzymes are going to be released, and these break down the pathogens. And then there is interference, which are non-specific responses to viral infections. In inflammation, here when damage occurs due to bacterial entry, presence of bacteria in the body is going to trigger the mast cells, which are found beneath the connective tissues of the skin, as well as around blood vessels, and the basophils to release histamines. These histamines cause the blood vessels to dilate. When they dilate, meaning more blood is going to come to that area, and this is going to lead to heating. Histamines are going to cause slight separation of blood vessels, and this is going to lead to the outflow of plasma towards that area, and all these together increase the temperature. The plasma that comes can contain antibodies, and these antibodies can disable the pathogen as the neutrophils and macrophages carry out phagocytosis. Here we can see what takes place when there is a cut around the skin. Some bacteria are going to enter the skin, and presence of bacteria is going to trigger the white blood cells called the mast cell to release histamines. The release of histamines by these blood cells increases permeability of blood vessels. We can see here, these are the histamines, the increased permeability of the blood vessels, and this is going to stimulate other immune cells to come into the area. The increased flow of blood is going to cause swelling, pain, as well as redness. And finally, the white blood cells, which are the neutrophils, are going to participate in phagocytosis in order to remove the threat or in order to remove the pathogens. Next, we move to fevers. When there is infection by a pathogen, it causes the hypothalamus to reset the core body temperature. Since most pathogens reproduce at about 37 degrees Celsius, a higher temperature is going to decrease the reproduction rate. The immune cells function better at higher temperature, but the pathogens are going to have a lower reproduction rate, and this is going to help the body to combat the infection. Usually, if there is a bacterial infection, the temperature rises steadily and remains high until the treatment can be successful. But for viral infections, the temperature spikes, and then it can go down, and then it spikes again. The spikes occur when the viruses break out of cells, and then this is going to decrease towards normal. Because bodies have enzyme-controlled reactions, if the temperature goes beyond 40, it could cause permanent tissue damage as well as death. Going on to phagocytosis, and again, this is still a non-specific immune response. This is carried out by neutrophils. During phagocytosis, digestive enzymes are going to be released from the lysosomes. These enzymes are called lysozymes. They are released from the vesicles, which are called the lysosomes, and they'll break down the pathogen. So both neutrophils and macrophages have lysosomes, but the lysosomes of the neutrophils cannot be regenerated or replenished. Once the enzymes are used up, they cannot digest more pathogens, and so they only ingest fewer pathogens before they die. Macrophages also participate in phagocytosis. They are usually monocytes, but when they move into the tissues, they become macrophages. And these macrophages ingest numerous amounts of pathogens because they can renew their lysosomes, so they last longer. When phagocytosis is done, the phagocyte produces cytokines, and these chemicals will stimulate phagocytes to move to the site of infection. This is going to raise body temperature, stimulating specific immune response. The last part of the non-specific immune response involves interference, which is the body's response to viral infections. When cells are invaded by viruses, they produce interferons, and these interferons are proteins that inhibit the replication of viruses within cells. The produced interferons can also diffuse out of the cells, and they could go to surrounding cells where they bind onto receptors. Of uninfected cells, they should be uninfected. Of uninfected cells, and this is going to stimulate pathways that cause those cells to be resistant to viral infections. It can signal neighboring cells to protect themselves. Some put on barriers to infection. Others are going to go through programmed cell death. And then if they're immune cells, they're going to be activated. Going on to topic 6b2, specific immune response to infection. Here the key characteristics of specific immune response include: it can distinguish self from non-self, and its specific meaning, it responds to only specific foreign antigens. It is diverse, meaning it can recognize more than 10 million different antigens, and it has immunological memory, of course talking about memory cells, meaning once it encounters a specific pathogen, it will respond faster on second encounter during secondary immune response. Lymphocytes and macrophages are the main white blood cells involved in specific immune response. These lymphocytes are made in the white bone marrow of long bones, and they move around the body in the blood as well as lymph, and they are involved in recognizing foreign antigens because, of course, they're going to produce antibodies. So the different kinds of lymphocytes include the B cells as well as the T cells. We're going to look at those later. These B cells are produced in the bone marrow, and they do mature in the lymph glands and more freely in the body. They have receptor proteins which are globular on their surfaces. These are going to be identical to the antibodies that they're going to produce later on. These antibodies are immunoglobulins because they are globular proteins involved in immune response, so we can call them immunoglobulins. When these B cells bind onto specific antigens, they lead to production of different types of B cells. The B cells could be effector cells, which can divide to form plasma cell clones. The plasma cells are going to produce antibodies for a particular antigen, and the B memory cells are the ones that will not produce antibodies at a time, but they will remain in the body with immunological memory to a specific antigen, so that on secondary infection, they can produce antibodies, or they can rapidly divide and produce many clones, which can produce antibodies faster in order to increase the concentration and fight against infection. The other kind of lymphocytes are the T cells. And again, T cells include the T killer cells, T helper cells, and T memory cells. So for the T cells, these are produced again in the bone marrow, and they mature in the thymus gland. They display thousands of identical T cell receptors. The T cells bind to antigens of infected body cells. When they do, this is going to trigger other types of T cells that play different roles. The T killer cells produce chemicals that destroy infected body cells. These chemicals are called perforins. And the T helper cells, these are very vital in specific immune response because they do activate the B cells to produce antibodies against a specific antigen to label for phagocytosis. The others are T memory cells that have immunological memory to a specific antigen. Beginning with a humoral response, all cells have specific molecules on their surfaces. So for pathogens, we can call these molecules antigens, and these can be detected by the body immune cells. So antigen presentation can take place by the body cells. And when antigen presentation occurs, the humoral response is going to lead to production of antibodies. But to produce antibodies, we have to activate T cells to produce T helper cells. And the activated T helper cells are going to cause the activation of B cells. And these B cells will then produce the antibodies, which carry out agglutination, neutralization, and so on. In the first step of the humoral response, we have the T helper cell activation stage. Here, there is going to be entry of a pathogen into the body, and some chemicals like histamines are going to be produced. These attract the macrophages as well as neutrophils. Remember, these are the ones that carry out phagocytosis. So these are going to engulf the pathogen in a shorter period of time, and they will destroy the pathogen. However, the antigens are going to be sorted out, and these are going to be attached or combined to the MHC protein, which is major histocompatibility complex, and these are going to be presented on the outside of the membrane of the macrophage. This is the process we call antigen presentation. So now we have an antigen presenting cell. So when the T cell binds onto this specific antigen presenting cell, this is going to trigger the T cell to produce clones of its own. One is going to be the T helper cell, and the other is the T memory cell, like we see here. So here we have a pathogen that is going to be engulfed by a macrophage. So we're going to have the antigens sorted out, combined onto the MHC, and exposed on the outside in the process of antigen presentation. So this is going to be the APC cell, or antigen presenting cell. Now this antigen presenting cell comes into contact with a T cell. This T cell is going to be activated if the two bind onto each other, because of course they have receptors that are complementary to that specific antigen. And when the T cell binds onto the antigen presenting cell, this T cell is going to go through rapid mitosis to produce two types of cells. Some are going to be the T memory cells, and the others are going to be the T helper cells. The T helper cells are going to be activated, and these are going to cause the activation of the B cells in the next stage, while the T memory cells will remain inactivated until secondary infection. So the effector stage, which is going to lead to the formation of the B memory cells as well as the B plasma cells. Some B cells, of course, also have proteins on their surface, and these are going to be compatible to the antigen on the pathogen. So the B cells are going to bind onto the pathogen, they will engulf it, and the fragments produced from the pathogen are going to be attached onto the MHC complex, and this is going to be presented. So we have a B cell that is an antigen presenting B cell. Remember, we already have the activated T helper cell from the previous stage, and this is going to recognize the antigen displayed on the MHC B cell, which is presenting the antigen. So it's going to bind onto it. When it binds onto it, this leads to production of cytokines, and the cytokines are going to cause the stimulation or the rapid division of the B cells, forming identical clones or multiple cells. Some of these are going to be B effector cells, and others are going to be the B memory cells. So the B memory cells will remain in the body for secondary infection. However, the B effector cells are going to differentiate and form B plasma clones or B plasma cells, and these cells are the ones that produce antibodies that carry out neutralization opsonization, and agglutination. Agglutination causes the pathogens to come together or more pathogens to stick to each other so that they are destroyed through phagocytosis. Opsonization is acting as markers. They label the pathogens so that the macrophages can recognize them. And then neutralization could be for toxins that are produced by the pathogen, as well as binding onto the pathogens so that they do not bind onto other body cells to cause infection. So here we have the bacterium. This is going to be the B cell. The B cell is going to bind onto the pathogen and it's going to become an antigen-presenting B cell. This antigen-presenting B cell is going to bind or is going to have a connection with a T helper cell that has been activated. And when that occurs, cytokines are going to be released. These cytokines cause the cell to go through rapid cell division, producing the B memory cells, as well as the B effector cells. Then these effector cells are going to differentiate to become B plasma cells, and those plasma cells are going to produce antibodies which will carry out opsonization, agglutination, and neutralization. The next phase is the cell-mediated response, in which we have the T killer cells. So this works on infected body cells, antigen-presenting cells, where antibodies cannot work. If an infected body cell digests a pathogen, it's going to carry out antigen presentation and it's going to be an antigen-presenting body cell. So these T killer cells also have receptors that are complementary to that of the pathogen. These are going to bind onto the MHC complex, and when these are exposed to the cytokines that were produced by the T helper cells, the T killer cells are going to go through rapid cell division and again producing two types of cells. One are going to be the T killer cells and the others are going to be the T killer memory cells. So these T killer cells are going to produce perforins, and these perforins are going to cause cells to have holes through their membranes or perforations, which cause excessive uptake of water by osmosis, causing those cells to swell and rupture. And when they do, the pathogens are going to be released. When these are released, the antibodies are going to mark them so that phagocytosis can occur. So here we have an infected body cell. This infected body cell is going to become an antigen-presenting cell. And when this antigen-presenting infected body cell meets a T killer cell, the T killer cell is also going to have a complementary receptor to the pathogen or to the antigen. So in the presence of cytokines which were produced by the T helper cell, this is going to go through rapid cell division, producing the T killer cells that are going to be activated, as well as the T killer memory cells. The activated T killer cells are going to produce perforins, and these perforins are going to poke holes into the cell membrane, causing the uptake of water excessively, and the cells are going to burst, releasing the pathogens that are trapped inside, and so antibodies are going to label them for phagocytosis by the macrophages. Now we move on to primary and secondary immune response. As you can see here, this is measured by the concentration of antibodies over time. In primary immune response or initial exposure, we can see it takes longer for the maximum concentration of antibodies to be attained, and actually the maximum attained is going to be less in comparison to the maximum attained on secondary exposure. So this means primary immune response takes longer and it's less effective because there were no memory cells. But because secondary immune response occurs when the memory cells are already in the body, and of course there is going to be some antibodies, these memory cells are going to rapidly divide. They'll produce antibodies within a shorter period of time, and the concentration is going to be more than that produced in the primary immune response. And that means sometimes the symptoms of disease may not even be felt, or they may not evolve. So in simple terms, secondary immune response is quicker and more efficient, and it produces higher concentrations of antibodies within a shorter period of time. Moving on to topic 6b3, developing immunity. Here we're going to look at the different types of immunity. These include active as well as passive. Active immunity involves formation of memory cells, but passive can only use maybe antibodies. Active immunity includes vaccination as well as actual infection, and these two both involve formation of memory cells. Passive immunity can include breastfeeding, so antibodies transferred from mother to child, as well as in blood transfusion where antibodies are transferred from some other organism or from another person. When vaccination is carried out, sometimes they use inactive viruses or pathogens. They can also use dead pathogens. They can use those that are weakened. and sometimes they just use an antigen like they do for influenza. And these days they can use DNA as well as RNA segments because these are going to lead to protein synthesis, which is going to lead to the production of maybe the antigen, which is going to be a protein, and that is going to trigger immune response in the body. Moving on to vaccination and herd immunity. Herd immunity can be achieved when a significant proportion of the population has been vaccinated against a specific disease, and this ensures that certain groups of people are protected, like those that cannot be vaccinated. This includes the babies, very old people, those who have compromised immune systems, and so many more. So if the biggest population is vaccinated, then those that cannot be vaccinated are not going to commonly come into close contact with sick individuals, and therefore the spread of that specific infection is going to be decreased, and that protects them from infection. Moving on to topic 6b4, treating bacterial disease. Bacterial diseases can be treated with antibiotics. Also, fungal diseases can be treated with antifungals. However, antibiotics have selective toxicity. Some antibiotics are bacteriostatic, while others are bactericidal. Bacteriostatics inhibit growth and reproduction of bacteria, while the others could completely destroy bacteria. And again, we can see how they work by looking at this. Some of these interrupt metabolic pathways by blocking maybe nucleic acid synthesis, meaning DNA won't be made, and if DNA is not made, then replication cannot occur and mitosis is going to be prevented. The others can inhibit protein synthesis by maybe preventing transcription as well as translation, and that is going to affect the cells in some way. Others can have connections with how the cell wall forms, preventing cross-linkages, and this is going to cause bacteria to take up water excessively, causing them to burst, and so many more. We have DNA gyrase inhibitors. These can stop DNA from winding into rings, and so it will not be able to fit into the bacterium, and therefore the cells will die. Moving on to topic 6b5, antibiotic resistance. Antibiotic resistance is one of the most common problems we are facing today because if pathogens are resistant to antibiotics, then it means the diseases that they cause could eventually kill many people. Most antibiotics are only effective if the microorganism has a binding site for the drug and a metabolic or biochemical pathway to interfere. Like we've seen here, all these antibiotics have a way they interfere in the growth and development of the pathogens. If mutations occur, the microorganism could gain advantage and therefore become resistant to the antibiotic. Some mutations can result in new biochemical pathways, and these may not be interfered by the antibiotic, and that means the pathogens will not be destroyed by the antibiotics. Sometimes these mutations can lead to different genes, and these genes could produce enzymes that can destroy the antibiotics, and that means they will not be effective. So there is a race between the pathogens and the scientists. That's why there are recommendations to stop the excessive use of antibiotics in order to minimize the exposure of these pathogens to the antibiotics, because the antibiotics act as selection pressure, and when these pathogens are exposed to this selection pressure, there is a chance that a random mutation could occur, and if this is advantageous, then they are going to survive and therefore become resistant to that specific antibiotic. Examples of drug-resistant bacteria include this MRSA, as well as Clostridium difficile. So it's recommended that antibiotics are only used when necessary, and people should understand the need to complete each dose of antibiotics. Also, using a few different antibiotics and keep some for reserve can be a solution in case others fail. And lastly, we look at controlling hospital-acquired infections, and again with the MRSAs. So people should wash hands and keep good hygiene practices. Patients should be isolated to prevent these infections from spreading. People should be tested before they come into the hospitals or before they are put in specific regions of the hospitals in order to prevent incoming infections. And then there should be bodies that monitor hospital-acquired infections in order to study the trends to see if these infections are increasing or they are decreasing so that a specific solution can be reached. So this brings us to the end of this topic. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.